New micro-injection immunotherapy destroys massive cancer and saves man's life. This is the story of a man whose science seemed to have left behind. A man told to go home, get his affairs in order, and prepare for the end. His name is John, and when doctors found the cancer inside him, it was already a monster. But this isn't a story about death. It's a story about refusing to accept the unacceptable. It's about a revolutionary approach to fighting cancer being explored by researchers, one that doesn't poison the whole body to heal it, but instead awakens its own hidden power. This is the story of how a single targeted injection of an experimental therapy began to do what months of toxic chemotherapy never could, shrinking a massive, inoperable tumor and rewriting a death sentence into a declaration of hope. My doctor looked me in the eye and gave me 6 months. The stage 4 tumor inside me was the size of a fist and growing. We hear these words, or variations of them, far too often. For John, the diagnosis was a stage 4 neuroendocrine carcinoma, a rare and notoriously aggressive cancer that had taken root in his liver. The scan was terrifying, a sprawling mass that was rapidly consuming his life. They told me my only option was chemo that would destroy my body, he remembers. I refused to spend my last days that way. And that refusal, that single act of defiance against a grim and predetermined path, changed everything. He asks, but what if there was another way? A revolutionary type of treatment that could shrink a massive tumor with just a single injection. This is my story. The problem with standard care and the wall of despair. To understand the miracle of what comes next, you first have to understand the brutal reality John was facing. When a patient is diagnosed with certain late-stage cancers, the path forward often looks depressingly similar. The primary weapon is systemic chemotherapy. Think of it like this. Imagine your tumor is a single venomous fish in a massive, vibrant coral reef. Standard chemotherapy is like pouring barrels of poison into the entire ocean, hoping enough of it reaches that one fish to kill it. The problem? The vast majority of the poison never hits its target. In fact, for many solid tumors, it's estimated that a very low percentage of the systemic chemotherapy drug actually penetrates the cancerous mass. The rest, that's the collateral damage. It can flood healthy organs, the nervous system, bone marrow, and most critically, the very immune cells that are your body's natural cancer fighters. The result is a litany of devastating side effects, crippling fatigue, nausea, hair loss, and a decimated quality of life. All while the tumor, shielded by its own unique defenses, often continues to adapt, resist, and grow. For John, this wasn't an abstract concept. It was a choice between two terrible ends, a fast death from the cancer or a slow, agonizing one from the cure. He saw friends who had walked that path, ravaged by the very treatments meant to save them. He and his family decided there had to be another way. His doctors, bound by standard protocols of care, had little else to offer. Surgery was deemed too risky. The tumor was enormous, and operating on the liver carries a high threat of complications. They had reached the end of their playbook. For many patients with advanced treatment-resistant cancers, the standard one-size-fits-all approach can unfortunately fail, leaving them exhausted, hopeless, and out of time. This is the wall so many patients hit, the moment when the medical establishment essentially says, there is nothing more we can do. It's a moment of profound isolation and fear. But for John, it was also a beginning. His refusal to accept this verdict sent him on a desperate search, an odyssey through the fringes of medical science, looking for a glimmer of hope. A new philosophy and a beacon of hope. That search led him to pioneering oncologists and researchers who are challenging the conventional wisdom of cancer treatment. Their central belief is radical yet simple. Your own immune system is the most powerful cancer-fighting weapon in existence. They argue that the standard model of care, with its overwhelming toxicity, can sometimes do more to suppress this weapon than unleash it. For years, specialized clinics and research centers have focused on a single mission: helping the thousands of patients who, like John, were told that nothing more could be done. When these researchers looked at John's case, they didn't just see a patient with a grim prognosis. They saw a patient whose own body held the key to his survival. They explained the core challenge in a way that finally made sense. Most aggressive tumors, a researcher might explain, are what we call immunologically cold. They've developed sophisticated cloaking mechanisms. They put up barriers and send out signals that make them invisible to the immune system. They're like spies hiding in plain sight. Systemic immunotherapies, like the checkpoint inhibitors you see advertised on television, have been a breakthrough. They work by taking the brakes off the immune system. But if the immune system can't see the tumor in the first place, releasing the brakes does nothing. This is why these incredible drugs often lead to long-term wins for a relatively small percentage of patients with these cold tumors. The question then becomes, how do you make a cold tumor hot? How do you strip away its disguise and paint a giant target on its back for the immune system to see? This is where John was introduced to a concept that would change his life: a precision-guided intratumoral immunotherapy. It's an approach that flips the conventional script on its head. Instead of poisoning the entire ocean to kill one fish, what if you could deliver a payload of immune-boosting medicine directly into the heart of the tumor itself? What if you could turn the tumor into its own cure? For John, who was running out of options and time, this was the leap of faith he was willing to take. He was crossing the threshold from the world of standard, accepted medicine into the pioneering and investigational world of next-generation oncology. The science of invisible surgery. The procedure is a marriage of interventional radiology and advanced immunotherapy. It's a form of invisible surgery that is both elegant in its precision and powerful in its potential effect. Here's how these experimental treatments generally work, broken down into two critical steps. Step 1. Advanced reconnaissance, multi-omics profiling. Before any treatment begins, you must first understand your enemy. Standard oncology typically relies on basic biomarkers and staging. But this approach, part of a growing field called precision oncology, goes infinitely deeper. It often involves what's called multi-omics testing. This isn't just a simple biopsy, it's a comprehensive intelligence report on the cancer. As a researcher might put it, imagine we're conducting a drone scan of the entire battlefield. We're not just looking at the tumor's location. We are mapping thousands of markers, its DNA, its RNA, its proteins, and its unique immune footprint. We're identifying exactly how your specific cancer is hiding and what its precise vulnerabilities are. This creates a personalized cancer portrait, a plan built for a single patient, not for a population statistic. The heartbreaking truth is that many patients may receive mismatched treatments based on generalized guidelines. This deep testing helps eliminate the guesswork. Step 2. The direct strike, the microinjection. Once the cancer's profile is understood, the procedure begins. It's minimally invasive. Under advanced 3D imaging guidance, a specialist navigates a catheter, sometimes as thin as a human hair, through the body's arteries directly to the blood supply feeding the tumor. There are no large incisions, no major surgery, just a tiny entry point, often in the groin or wrist. Once the catheter is in place, the true innovation begins. The first part of the injection may use specific agents to help break through the tumor's dense, fibrous matrix. This physical disruption does something critically important. It can force the tumor to release its hidden antigens, the unique protein markers that act as cancer's ID badges. It also releases something called DAMPs, or damage-associated molecular patterns, which are essentially cellular alarm bells that scream danger to the immune system. Immediately following this, a custom-built cocktail of medications is microdosed directly into this newly exposed and chaotic tumor environment. This isn't a sledgehammer of high-dose chemo. It's often a precisely formulated mix of low-dose chemotherapies and powerful immune-stimulating drugs like cytokines and TLR agonists. This cocktail does three things simultaneously. 1. It kills a portion of the cancer cells directly. further spilling their identifying antigens. 2. It helps strip away the tumor's immunosuppressive cloak, making it visible. But most importantly, it acts as a clarion call to the immune system's first responders, the dendritic cells. These dendritic cells are the detectives of the immune system. They rush to the site of this commotion, gobble up the newly released cancer antigens, and race to the body's lymph nodes. There, they present this mugshot of the cancer to the immune system's elite assassins, the T cells and natural killer cells. And this is the holy grail of the treatment, the abscopal effect. By treating one tumor locally, you are potentially training the entire immune system to recognize and hunt down that cancer anywhere it exists in the body. The injected tumor effectively becomes an in situ or in-place cancer vaccine, one created from the patient's own unique cancer cells. This is how a local treatment can produce a systemic body-wide response, often without the systemic toxicity. You're not just killing one tumor, you're teaching the body how to kill them all. The treatment and the astonishing result. For John, the day of his first treatment was surreal. He had braced himself for the sterile, intimidating environment of a surgery ward or a chemo infusion center. Instead, the procedure was calm and focused. He was awake the entire time, feeling little more than the slight pressure of the catheter's insertion. Within a few hours, it was over. A single bandage was placed over the entry point, and he was able to go home. No hospital stay, no debilitating sickness. Then came the wait, the agonizing, hope-filled, terrifying wait. Had it worked? Could a single, minimally invasive procedure truly make a difference against a monster that was supposed to kill him in months? He and his family held their breath, praying for a miracle. Weeks later, they were back in the doctor's office to review the follow-up scans. This was the moment of truth. The doctor pulled up the images on his screen. On one side, the before scan showed the massive, sprawling tumor. On the other side was the after scan. The room fell silent. In cases like John's, which inspired this story, some patients in early clinical trials have seen remarkable responses. The tumor that had once dominated the scan showed significant shrinkage. With just one treatment, the massive growth was substantially reduced in volume. Tears streamed down his face. His wife gripped his hand, sobbing with a relief so profound it was almost painful. The seemingly impossible had happened. But the good news didn't stop there. The advanced testing had provided other crucial metrics to track progress. One of the most important is circulating tumor cells, or CTCs. These are cancer cells that break off from the primary tumor and travel through the bloodstream, acting as seeds for metastasis. A high count is often associated with a worse prognosis. After the treatment, a patient might see their CTC count drop dramatically, in some cases to 0. Another key set of metrics can come from deep molecular profiling, which measures the genetic and epigenetic changes that drive a cancer's aggressiveness. After treatment, tests might show that the cancer's engine of growth had been fundamentally crippled. And perhaps most tellingly, there was the impact on his quality of life. Before the procedure, John's days were marked by pain and fatigue. Within weeks, he didn't just have scans that looked better, he felt alive again. He had energy. The constant pain began to subside. He could play with his grandkids. He was living, not just surviving. Pioneering researchers note that this kind of result, while remarkable, is part of a pattern they are working to confirm in clinical trials. John's story was a powerful example of what precision, immune-focused oncology hopes to achieve. A new beginning and a message of hope. John's journey is far from over. Cancer is a relentless adversary. But for the first time in a long time, he is fighting on his own terms. His life is no longer measured in months, but in moments. A walk in the park, a family dinner, the simple joy of waking up without debilitating pain. These are the victories that matter most. His story is a powerful testament to a paradigm shift that is slowly but surely happening in oncology. The future of cancer care isn't just about finding stronger poisons, it's about becoming smarter. It's about recognizing that the most sophisticated pharmacy in the world exists within each and every one of us. Approaches like direct-to-tumor immunotherapy are at the forefront of this revolution. They are still considered investigational by many, and hundreds of clinical trials are underway to validate their effectiveness across different cancer types. But for patients like John, who may have exhausted standard options, they represent a vital lifeline. This story is not just for inspiration. It's a call to action for empowerment. If you or a loved one is fighting a battle with cancer, especially if you've been told your options are limited, it's time to ask different questions. Don't settle for standard of care as the only answer. Go to your doctor, to your oncologist, and ask them today, are there any clinical trials for intratumoral immunotherapy that I might be a candidate for? Could a referral to an interventional radiologist or oncologist be an option to explore localized treatment strategies? Are we doing advanced testing? Can we get a multi-omics profile to map out my cancer's unique vulnerabilities and see if it opens up new personalized therapy options? Can we test for circulating tumor cells, CTCs, or cell-free DNA to get a real-time indicator of my cancer's activity? If the answer is no, or if they are unfamiliar with these concepts, seek out cancer centers and researchers that are. This is the new frontier of precision oncology, safer, more targeted, and powered by your own biology. You have fought this hard to get here. Now is the time to fight smarter. The journey to healing is long, but it always begins with hope. John's story is proof that even in the face of a terrifying diagnosis, there are new reasons to be hopeful. Science is making the impossible possible. If this story sparked hope in you, please consider subscribing. Your click helps push this life-changing message out to more patients, more families, and helps build awareness with the institutions that can make these treatments more accessible. Share this video with someone you know who is fighting this battle, because healing always starts with hope. May you find strength on your journey.